Hello friends, good afternoon and welcome to Reset Live Lectures. Dear friends, as you know that we have started a series on developmental biology and today we are conducting yet another lecture in the same series. In today's lecture, we will discuss about implantation and placentation. In this lecture, we will try and understand implantation of the embryo in the uterus of the mother. To discuss this topic, we have with us our sub-expert Dr. Charu Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you. So we have seen during the ovulation, the egg is released from the ovary and taken up by the fallopian tube. There is a special part at fallopian tube where the fertilization can take place. If the sperm has entered into the female's reproductive tract, the sperms travel up to the upper part of the fallopian tube and there the fertilization can occur. Now after the fertilization has occurred and the sperm and egg has fused together to form the zygote, the further development to form an embryo needs to take place. Now for that it needs a conducive environment, and that environment is provided by the uterine epithelium. The moving zygote or the rolling zygote down the fallopian tube comes into the uterus and implants itself or embeds itself into the uterine wall called as the uterine epithelium or endometrium, and the further development takes place. So this is the implantation which we will talk about in today's session. And then the growing embryo needs to take up different functions such as breathing, the respiration, the nutrition, the excretion, and for that it makes a connection with the maternal tissues. And the process of formation of this fetal and maternal exchanging system is termed as the placentation, and that structure is termed as the placenta. So the specific objectives of this particular session would be to understand the mechanism of implantation, and we will also look at the certain anomalies, the form anomalies and implantation to go about. So as I said, implantation is a process in which a developing embryo moving as a blastocyst through a uterus makes contact with the uterine wall and remains attached to it until birth, when it separates from the endometrium, a phenomena termed as parturition, and the baby birth takes place. So implantation is when it embeds itself into the uterine wall for the further development to take place. It is the final frontier to embryogenesis and successful pregnancy. In humans, there is a precise coordinated timeline in which the pregnancy can occur in the uterus, and this is called as the window of implantation. So before we talk about how the implantation takes place, let's review the moving zygote in human case, which is termed as the blastocyst, and let's look at what makeup is of the endometrium when the implantation needs to occur. The developing human embryo is the blastocyst. The blastocyst is a hallmark of mammalian cleavage. Due to the phenomena of compaction, the blastomeres following the third cleavage huddle together, maximizing their contact with one another and forming a compact ball of cells. So when the cleavage is taking place, a peculiar phenomena called the compaction occurs in human development when the blastomeres just huddle together and form a compact ball of cells. The 16-cell morula consists of a small group of internal cells surrounded by a larger group of external cells. Most of the descendants of the external cells become the trophoblast or the trophoectodermal cells, the group of cells that produces no embryonic structures but forms the tissue of the extraembryonic membrane called the chorion. So that's the division that takes place. The embryo proper is derived from the descendants of the inner cells of the 16-cell stage. These cells generate what is termed as the inner cell mass, which will give rise to the embryo and the associated extraembryonic membranes, the allantois, the yolk sac and the amnion. So this is how the cleavage takes place, and this is the structure which is termed as the blastocyst. So the compaction has occurred and there is an inner layer of cells termed the inner cell mass, and there is an outer layer of cells termed the trophoectoderm or trophoblast cells. During the process of cavitation, The trophoblast cells secrete fluid into the morula to create a blastocele. The inner cell mass is positioned on one side of the ring of the trophoblast cells, resulting in the structure termed as blastocyst. So, morula does not contain any cavity, but when the trophoblast layer secretes some fluid, there is cavitation that occurs and there is a blastocele that appears, so that the inner cell mass is pushed towards the one side. This structure is peculiar to the mammalian cleavage and it is termed as the blastocyst. Now, let us have a look at the uterine epithelium or the endometrium. The endometrium is a multi-layered dynamic organ overlaying the myometrium and comprises of a functional layer and a basal layer. So, you have a functional layer, you have a basal layer, and then there is a myometrium or the muscular tissue which is there. So, this entire region, including this functionalis and basilis, is termed as the endometrial layer. Each month cells in the functional layer are separated from the basal layer during menstruation. So, there is a developmental changes. The endometrium is a dynamic structure. It keeps on changing and it follows a certain cycle. So, every month the cells of the functional layer are separated from the basal layer and they are thrown out. The basal layer is attached to the myometrium and it remains intact during menstruation, serving as a base for endometrial regeneration. So, you can see here that this is functionalis, this is basilis and this is myometrium. So, basilis remains throughout. However, the functionalis layer, when this is the menstrual phase, then this is slowed off and this is released out of the body and the blood released out of the body, and then again it regains and undergoes a proliferative stage. During this proliferative stage, this stratum functionalis is again formed. Then there are uterine glands that develop. This is the time of ovulation when the ovum is released from the ovary, and then you can see that this is the secretory phase. It secretes a lot of substances, and after that, when there is a premenstrual phase, then through this the structure again kind of deteriorates, and during the menstrual phase this is again shed off. So, throughout the fertile period of women's life, hormonal changes control the cyclical changes in the reproductive organs. Every month an egg is released by ovulation from the ovaries and the endometrium thickens and so as to become ready for accepting the fertilized egg. If the eggs get fertilized, then it gets embedded here or implanted here. If it does not, then if conception does not take place, then this uterine lining is discarded and a menstrual bleed occurs. So, endometrium wall by itself under the hormonal changes undergoes this kind of a cycle, and therefore it is a very dynamic structure. The endometrium is composed of several different cell types, including luminal and glandular epithelial cells, stroma with stromal fibroblastic cells, immunocompetent cells and blood vessels. We'll have a look at the hormones which play a part. So, we'll talk about the endocrine mediators and we'll talk about certain other effector molecules which affect this endometrial cycle. So, progesterone and estrogen are the dominant hormone modulators of endometrial development. Ovarian estrogen and progesterone condition the uterus for implantation, such as thickening the uterine wall. Both the epithelial and stromal compartments express progesterone and estrogen receptors, and the response depends on the levels of these receptors as well as on the concentration of the hormones themselves. So, concentration of these hormones depend upon the state of the body, and moreover the concentration of the receptors of progesterone and estrogen is also dynamic. So, based upon the concentration of both these, the epithelial and stromal components, they are going to change. The hormonal activity also depends upon the rates of the progesterone and estrogen metabolism. For example, upregulation of enzymes that convert estradiol to estron or estron sulfate or remove the sulfate from E2 and estron. So, basically not only the concentration of the hormones which is making a difference, but also the metabolism of these hormones make a difference for the endometrial lining. The activities of progesterone and estrogen are also influenced by the effects of certain coactivators and repressors. Furthermore, both hormones regulate the expression of numerous endometrial proteins by paracrine signaling. In addition to progesterone and estrogen, a number of other endocrinological factors such as prostaglandins in rodents are known to mediate endometrial function. Endometrial factors are also critical mediators of all phases of the implantation process. So, this is the role of progesterone and estrogen receptors during endometrial development. So, you can see, when the ovulation is taking place, and we have already seen the growth of the endometrium, and then, finally flowing of the endometrium functionalis, the stratum functionalis during the menstrual period. So, this is how basically it starts. The E2 stimulates the expression and the proliferation, and then the proliferation occurs, and then the proliferation ceases, the progesterone blockade of ER, and then increase in the other activities increases the E2 to E3 conversion, then increased secretion of glycoproteins and peptides into the uterine cavity. And you can see then this is the maximal growth and the maximal, the kind of vascularization that has occurred. The peak secretory activity, decidulization, it still remains in the circular form. The progesterone dependent infiltration of specific leukocyte subsets which increased mitosis and differentiation in the stroma. And then further down, when the levels decreases, then this layer is slowed off and shed. So, this is how the E2, E3, the progesterone and the estrogen as well as the estrogen receptors play a role during the endometrial development. And as I said, there are other factors that regulate during the early stages of implantation. For example, in the trophoblast layer of the blastocyst, the factors such as adhesion molecules, and we will look at the cytokine and chemokine receptors, and then there are certain proteases which are there. Then in the luminal epithelium, the factors that play a role are the glycocalyx components, again the adhesion molecules, cytokines, chemokines, proteases, and certain calcium regulators. Then in the glands, again the growth factors and the nutrients also play a major role. Then in the extracellular matrix, the hyaluronan, the collagens, and the degradative enzymes inhibitors. And in the decidulizing stroma, there are prostaglandins and other cytokines and chemokines and proteases, that factors which are regulated. So, all the changes in the endometrial wall that occurs, that leads to the conducive environment for implantation, these are the factors that play a role in successful implantation or early stages of implantation. So, when the blastocyst is traveling down the fallopian tube, it is encased inside the outer layer which is called as the zona pellucida. Now, it has to escape from the zona pellucida to be ready to be implanted in the uterus. So, while the embryo is moving through the oviduct en route to the uterus, the blastocyst expands within the zona pellucida. The plasma membranes of the trophoblast cells contain a sodium pump, a sodium potassium ATPase, facing the blastocele, and these proteins pump sodium ions into the central cavity. This accumulation of sodium ions draws in water osmotically, thus enlarging the blastocele. So, the blastocele keeps on expanding, and therefore, the growing blastocyst or the growing embryo inside the zona pellucida also keeps on expanding. Then there are certain kind of proteases, just like trypsin. They make a hole. For example, it has been studied in the rodents that they make a hole in the zona pellucida, and the entire embryo, the growing embryo, just squeezes out of the zona pellucida, and this phenomena is termed as the hatching. So, what happens is the mouse blastocyst hatches from the zona pellucida by lysing a small hole in it and squeezing through the hole as the blastocyst expands. A trypsin-like protease, which is termed as the trypsin, is located on the trophoblast cell membranes, and it lyses a hole in the fibrillar matrix of the zona. Once out, the blastocyst can make direct contact with uterus. So, this hatching is very important. The zona pellucida prevents the implantation of uterus at any other portions apart from the uterus. So, while this blastocele is transporting from the fallopian tube, It does not allow the blastocyst to implant at the fallopian tube, at any part of the fallopian tube or at any part of the uterus apart from where the implantation needs to take place. So, hatching out from the zona pellucida makes sure or leads to the next step that now the blastocyst can be implanted into the uterine wall. So, now we will talk about the different the mechanism of implantation or different phases of human embryo implantation. The human embryo enters the uterine cavity approximately 4 to 5 days post fertilization. After passing down the fallopian tube, the embryo is moved within the uterine lumen by rheumatic myometrial contractions until it can physically attach itself to the endometrial epithelium. So, basically the blastocyst is under a stress condition. These mucin and the myometrial contractions is creating a kind of stress, and this shear stress it has to withstand, and moreover under that stress it needs to implant into the uterus. It hatches from the zona pellucida, as we have seen, within 1 to 2 days after entering the cavity, thereby exposing the trophoblastic cells of the trophoectoderm to the uterine epithelium. Implantation occurs 6 to 7 days after fertilization, and this interactive process is a complex series of events that can be divided into 3 distinct steps. We will see the apposition, where basically the blastocyst orient itself and just lie next to the uterine epithelium. The attachment, when it actually attaches to the endometrial wall, and then invasion, when the growing trophoblast, the kind of trophoblast basically invades, and it invades the maternal cell wall and the maternal wall, the endometrial wall, and basically it then gets inside the endometrial epithelium, and the epithelium actually grows over it, and then it is said to be transplanted and the growth occurs. So, we will talk about these 3 mechanisms, the 3 phases: the apposition, the attachment and the invasion part. So, apposition step is a transient and dynamic process whereby the embryo tethers itself to the endometrial surface. Despite uterine contractions and mucin secretions within the uterine cavity, which as I said creates a kind of shear stress, the embryo is still able to approach the wall, roll around to right itself so that the trophoectoderm overlying the inner cell mass opposes to the endometrial surface. So, basically the trophoectoderm layer, which is just which is just outside the inner cell mass, it orients itself in that manner that part where the inner cell mass and then this trophoectoderm layer is there lies right next to the uterine wall, or it just tethers or it opposes itself onto the uterine wall or endometrial wall which is there. During this apposition phase, there is a dialogue between the floating blastocyst and endometrium using soluble mediators such as cytokines and chemokines, acting in a bidirectional fashion to guide the blastocyst onto a 3-dimensional docking structure. So, there is a lot of effector molecules which play a role, and finally it assumes a proper orientation in the docking structure. The hormone regulated pinopods at the endometrial surface have been shown to mark the timing and appearance of optimal endometrial receptivity. As I said, there is a window of implantation, and that small window of implantation is when the blastocyst opposes itself correctly towards the endometrial membrane, and when the attachment needs to occur and invasion needs to start. So, that small membrane, everything needs to go in order, and then only the successful implantation takes place. The chemokines such as IL-8, Rantis or MCP1 and the L-selectin system in particular has been shown to be critically important during the blastocyst apposition phase. Studies suggest that the human embryo uses a mechanism well studied in leukocytes to mediate rolling and tethering onto the endometrial wall prior to form the adhesion. So, just like leukocytes kind of infiltrates through the blood vessels, and the way they work, has been in a similar mechanism, the blastocyst or the human embryo apposition also takes place. The next phase is the embryo needs to attach to the uterine wall. Shortly after the apposition step, integrin dependent adhesion and attachment occurs. This receptor mediated event allows the blastocyst to attach firmly to the uterine wall, and trophoblasts transmigrate across the luminal epithelium, burying the embryo beneath the uterine wall. So, in embryo attachment, basically the receptor mediated event allows, so integrin dependent, so integrins play an important role. These are the cell adhesion molecules, and they cause the blastocyst, the moving blastocyst, to actually adhere to the or attach itself to the endometrial wall. And as you can see, this is the portion when it tries to oppose itself, and this is the part wherein these are the pinopodia, the hormonal secreting pinopods, and this is the time when actually the growing blastocyst, so it has orient itself, so the inner cell mass, and these are the outer cell trophoblast layer, and then these are the receptor molecules, and then they get attached in here, and then you can see that the attachment has occurred. So this is kind of apposition, and this is the attachment phase that has occurred. After the human blastocyst tethers to and breaks through the glycocalyx barrier, it then induces by paracrine crosstalk a favorable epithelial integrin pattern for its firm attachment. So initially it is loosely attached, and then it becomes firmly attached because of the paracrine signaling and the integral expression pattern that takes place. The blastocyst firmly gets attached to the endometrial wall. Then comes the phase of trophoblast invasion. The final phase of implantation is invasion, which permits the creation of the hemochorial placenta. So, we will talk about the various types of placenta in a subsequent session, but hemochorial placenta is basically when there is an interdigitation that has occurred, and there is an intermingling of the blood vessels which are there. So, there forms a structure where the fetal membranes, fetal vessels and the maternal vessels, they lie next to each other, and therefore the diffusion can take place, and this structure is called as the placenta. So, hemochorial placenta is basically develops when there is a strong interdigitation and the membranes are dissolved for the maternal one, and basically the fetal structures, they just kind of bathes in the maternal blood, and this permits the creation of such kind of a placenta. Invasiveness implies the ability of cells to cross the anatomic barriers. The first barrier is the layer of endometrial epithelial cells upon which the trophoblasts are attached. Immediately beneath the epithelial layer of cells is a specialized type of matrix which is called as the basement membrane, a thin continuous layer composed mainly of the type 4 collagen. Beyond the basement membrane lies a highly variable matrix called the interstitial stroma, which contains other tissues, cells, vessels and lymphatic channels. So, this is the the arrangement of the endometrial cells. So, superficially there are the endometrial epithelial cells which are lying. Underlying there is a matrix, a basement membrane, and under it there is a interstitial stroma which contains other tissue cells such as vessels and lymphatic channels. Since the blastocyst is too large to squeeze through the epithelial layer, the attached trophoblast use paracrine activity to induce an apoptotic reaction in the underlying endometrial epithelial cell. So, it does release certain proteases which digest the ECM, and moreover there are certain molecules which basically induce the production of the signaling mechanism that causes the endometrial wall to be degraded, and therefore the blastocyst can be implanted successfully. To achieve successful invasion, trophoblasts then differentiate in a tightly regulated manner, producing anchoring cytotrophoblast and highly secretory syncytiotrophoblasts that degrades the extracellular matrix with several proteases. So when this implantation is taking place, the trophoblast layer actually differentiated into two different types, which is basically the syncytial trophoblast and the cytotrophoblast. So syncytial trophoblast basically it degrades the extracellular matrix by releasing certain proteases, and cytotrophoblast is basically for the anchoring of the blastocysts onto the endometrial wall. So this is where the invasion has occurred. So this is apposition, this is attachment, and this is where the invasion has occurred, and there you can see this is the cytotrophoblast and this is the syncytial trophoblast that has occurred, that is there. So cytotrophoblast causes the anchoring of the blastocyst onto the endometrial wall, whereas syncytial trophoblast basically releases various proteases that degrades the endometrial wall and causes the blastocyst to be embedded inside, and then this endometrial wall regrows over and above, and further development takes place inside this endometrial wall after the implantation has occurred. So there are certain kind of implantation and form anomalies which are there. For example, the placenta previa is characterized by a low-lying insertion location in the uterus which is close to the cervix. The natural birth canal can be partially or completely plugged up thereby and represent a mechanical obstacle during delivery through vagina. This pathology affects 0.5% of all the pregnant women. The latter risks hemorrhages due to the detachment of the placenta that occurs too early during uterine contractions or cervix dilation. So this condition is termed as placenta previa, where the implantation has occurred very lower down into the uterus, just above the cervix, and it causes the placenta to be formed there, and therefore the placenta just kind of it causes a lot of hemorrhage because the placenta can be taken off very early during the uterine contractions and cervical dilation for the parturition or the childbirth to take place. Then there is also ectopic, that is extrauterine pregnancies. This occur in approximately 1 woman in 300. They arise through an implantation of the embryo at another location than in the uterine cavity. The most frequent location is that of the fallopian tube. So as we have seen that the blastocyst needs to escape from the zona pellucida before being implanted. So zona pellucida is actually preventing the blastocyst to be implanted anywhere aside, but sometimes it happens that the premature escape can occur and the implantation occurs in, for example, at the fallopian tube, and this is termed as the ectopic pregnancy. Implantations in the ovary, in the abdominal cavity or the intrauterine parts of the fallopian tube can also ensue. So apart from that, implantations can also take place in the ovary or in the abdominal cavity, or intrauterine parts of the fallopian tube can also occur. So basically these are termed as the ectopic pregnancies, and it does not lead to the further development of the embryo. Coming to the placentation, by the 10th day after fertilization, the blastocyst is completely embedded in the stromal tissue of the endometrium, with the epithelium regrows over to convert the site of implantation. Eventually, the cytotrophoblast invade the entire endometrium, the uterine vasculature, and into the inner third of the myometrium. The access to the uterine vessels create the lacunar networks comprising the uteroplacental circulation, which places the placental trophoblast in direct contact with the maternal blood. So after the implantation has occurred, by the 10th day after fertilization, then the cytotrophoblast, they invade the entire endometrium and they'll come to lie under, they almost reaches the inner third of the myometrium. And then the access to the uterine vessel creates these kind of lacunar networks. And as I said that, the fetal tissue is basically bathing in the maternal blood, and the extraembryonic mesoderm then joins the trophoblastic extensions and give rise to the blood vessels that carry the nutrients from the mother to the embryo. The narrow connecting stalk of the extraembryonic mesoderm that links the embryo to the trophoblast eventually forms the vessels of the umbilical cord. the fully developed extraembryonic organ consisting of trophoblast tissue, and the blood vessel containing mesoderm is called the chorion, and it fuses with the uterine wall to create what is termed as the placenta. So this is how the implantation has taken place. This is how it is, the invasion part is going on. Then by the 10th day, the entire blastocyst has gone inside the endometrial wall and it has regrown. And as you can see, these are the trophoblastic lacunae which are there. And then this occurs that these are the maternal blood supply, the lacunae has also occurred, and this is the tissue and this is the growing embryo that is developing, and here the formation of the placenta occurs. And this looks like this. The placenta has both the maternal portion, the uterine endometrium which is modified. So this is from the mother side. So this is to the mother and this is from the mother, the blood vessels which have come, and this is the placental portion. And in placental portion there are a lot of villi which are there where the juxtaposition of the vessels takes place, and this is then it goes to the through the umbilical cord to pass on the material or to take the waste materials from the fetus. So this region is basically called as the placenta, which contains the portion from the maternal side and which also contains the chorionic portions from the fetal side. Although fetal and maternal circulatory system normally never merge, diffusion of soluble substances can occur through the villi which is there. So the blastocyst has now implanted into the uterine wall, the mother's uterine wall, and then there occurs the creation of a structure termed as the placenta, from where the exchange of materials between the maternal and the fetal tissue takes place. So the learning objectives for this particular session will be to understand more about the composition of this placental barrier, to understand the different types of placenta, and to understand the physiology of placenta. So this is the temporary organ that developed, that joins the mother and the fetus, and as you can see, this is the diagram of it. This is the umbilical cord where the vessels from the where the blood vessels, which is the connection between the growing embryo and the on the maternal side, and so this is the umbilical cord, and this is the amnion. This is the chorionic plate, as we have seen. So these are the extraembryonic membranes. So this umbilical cord connects to the embryo proper, and these are the fetal tissues again. So this is a fetal tissue, the amnion. This is the chorionic plate, and then this is the intervillous space where the maternal blood is there, and then there is a basal plate which is there. This is the basal plate. Each one is called as the cotyledon. It's not a complete cotyledonary placenta, the human placenta, but this one structure is kind as the cotyledon, and 7, these are all villi which are present. So the surface area of the exchange is basically enhanced because of the formation of these villi. Because the membrane is thrown into the villi, these villi increases the surface area where the exchanges can occur. So this is a schematic diagram of the placenta around the 4th month in the sagittal section, which is shown here. The fetal part of the placenta, as I said, is made up of the chorionic plate with its placental villi, the cytotrophoblast layer and the intervillous space. The chorionic plate, great part of the placenta on the fetal side, consists of the amnion, the extraembryonic mesenchyme, the cytotrophoblast and the syncytiotrophoblast, as we have seen. So, this is the chorionic plate which is there. So, this is the amnion, this is the extraembryonic membrane space, this is the mesoblast which is there, and these are the cytotrophoblast and the syncytiotrophoblast. The basal plate, the peripheral region of the placenta, which is basically present on the maternal side, that is in contact with the uterine wall. So, this is the uterine wall which is there, and they compose of two tissues, the embryonic tissue, which is basically the cytotrophoblast and the syncytiotrophoblast. So, here you can see this is the cytotrophoblast and syncytiotrophoblast which is there. So, it touches that on the one side, and of the maternal tissue, the decidua basalis on the other side. So, you can see this is the zona compacta, spongiosa, decidua basalis, and this is the myometrium which was present. So, this is the myometrium, this is the endometrial layer which we have already seen, and endometrial layer, so we have seen this is the functional layer and this is the basalis layer which we were talking about. So, in the functional layer, basically this kind of this is the maternal tissue composition of the placenta that has occurred. So, the chorionic plate etc. is basically given by the fetal part, and this part is given by the maternal part which is there. So, the placental barrier is composed of structures that separate the maternal and the fetal blood. The makeup of the placental barrier changes over the course of the pregnancy. In the first trimester, it consists of the syncytiotrophoblast layer, the cytotrophoblast layer, the villus mesenchyme, in which numerous ovoid Hofbauer cells are there that exhibit macrophage properties are also found, and the fetal capillary wall. So, this is the first trimester or within first 3 months of the conception schematic diagram, where you can see that this is the placental barrier composition which is occurring. So, this is the intervillous space, this is the syncytiotrophoblast, this is the cytotrophoblast, then there are the villi which are present, and these are the macrophage cells which are roaming around, and these are the fetal capillaries which are there. So, these are the macrophages and these are the fetal capillaries which are also present. Now, during the 4th month, the cytotrophoblast disappears from the serous wall and the thickness of the barrier decreases. So, you can see that this was actually the barrier between the intervillous space and this area. So, you can see now the syncytiotrophoblast has been disappeared and the thickness of the barrier has decreased, with the surface area increases roughly 12 meters square towards the end of the pregnancy. In the 5th month, the fetal vessels have multiplied their branches and gotten closer to the villus surface. You can see they are moving now towards the villus surface, and the branches have been multiplied. So, this now act as the placental barrier. Now, during the 6th month, the nuclei of the syncytiotrophoblast group together in the so-called proliferation knots. So, you can see that now these are the kind of the placental barrier which is there. This is again the intervillous space with the maternal blood. This is the placental barrier of a terminal villus, and within that this is the fetal capillaries which are there. So, this is the maternal blood, this is the fetal capillaries which are there, and this is the barrier which is occurring, and it composes of the merged basal membrane of the fetal capillary and of the syncytiotrophoblast. Then it contains the endothelial cells which are there. It contains rare cytotrophoblast cells. So, cytotrophoblast cells have also disappeared, but there are rare cells which are also there. Then basal membrane of the capillary. So, this is the basal membrane of the capillary. So, this complete is the fetal capillary. This is the maternal blood vessel. So, this is how and this is the placental barrier which is there. Then you can see this is the basal membrane of the trophoblast portion, and this is basically the syncytiotrophoblast with proliferation knots, the nuclei rich region. So, this is the nuclei, and this entire region act as the placental barrier between the fetal capillaries and the maternal blood wall that has taken place, and this occurs around the 6th month initially. So, it's again a placenta is a dynamic structure, and the placental barrier also keeps on changing as the development occurs. Now we will look at the different types of placenta. So based on the outer form, the morphological or the structural look of the outer morphology, the different types of placenta are like the diffuse, which is basically almost the entire surface of the allantochorion is involved in formation of the placenta. So we are talking about the allantochorion placentas. Basically in this case, the entire surface of the chorion allantoic is in the formation of the placenta, and this can be seen in the horses and the pigs. So this is the swine placenta which is shown here. So it covers the entire surface of the embryo. Then there is cotyledonary. So these are the cotyledonary placenta. Multiple discrete areas of attachments called the cotyledons are formed by interaction of patches of the allantochorion with the endometrium. The fetal portions of this type of placenta are called cotyledons. The maternal contact sites, which are called as the caruncles, and the cotyledon caruncle complex is a placentome. This type of placentation is observed in ruminants. So you can see this kind of placentation occurs in cows, and this is called as the cotyledonary placenta. Then there is a zonary placenta. The placenta takes the form of a complete or incomplete band of tissue surrounding the fetus. So this is the growing fetus, and here you can see that this zone is basically the placenta. It is seen in the carnivores like dogs and cats, seals, bears and elephants. So this is a zonary kind of a placenta which is there, and then there is a discoidal placenta. A single placenta is formed, and basically it's in the discoid shape as in seen in primates and rodents including the humans. So it is called a discoidal placenta, where the placenta still occurs like a disc. So this is the growing blastocyst, and this is the entire area, and this is appearing as a disc. So disc where the discoidal, where the disc-like, this is zone where this is the growing embryo, and this is the placenta is appearing in as a zone, and then there is a cotyledonary placenta, and then there is a diffuse placenta. So based on the morphology of the placenta, these four kinds of placenta occurs. Then based upon the feto-maternal interdigitation, these are the three layers of fetal extraembryonic membranes in the chorioallantoic placenta of all mammals, all of which are components of the mature placenta. So basically when there is a fetal and maternal proximity of the tissues, then there are three layers which are contributed by the fetal tissues, and there are three layers which can be distinguished from the maternal tissues. So in all, a proper chorioallantoic placenta has three layers where three are fetal, has six layers where three are fetal and three are maternal. The three fetal layers are basically the endothelium lining of the allantoic capillaries, the connective tissue in the form of the chorioallantoic mesoderm, and the chorionic epithelium, the outermost layer of fetal membranes, which is derived from the trophoblast. So you can see this is the fetal site. So there is a chorionic epithelial cells, there is a fetal connective tissue, and then these are the fetal endothelial cells which are basically present for the allantoic capillaries which are there. Then there are three layers from the maternal side also, and these maternal cells are the endothelium lining the endometrial blood vessels. So these are the endothelium lining the endometrial blood vessels. Then there is a connective tissue of the endometrium, and then there is the endometrial epithelial cells. So the chorionic epithelial cells are derived from the trophoblastic cell layer of the blastocyst, and then this is the endometrial epithelial superficial cells, endometrial cells which are there. So endometrial epithelial cells, so these are the six layers basically which forms a chorioallantoic placenta. Now depending upon which layers are present and which layers are degraded of the maternal side, because when this is like a juxtaposition, they are just lying adjacent to each other, but when it starts invading into the uterine wall, then this tissue will keeps on degrading, and therefore we have different histologically we have different kinds of placenta. So in these types of placenta, there is an epitheliochorial placenta, there is endotheliochorial placenta, and there is a hemochorial placenta. So what happens is, these are the maternal layers. So in the epitheliochorial placenta, all the three maternal layers are retained. So all the six layers are present. all the three layers are retained. So there is an endometrial epithelium, there is a connective tissue, and there is a uterine endothelium which is all intact. And this kind is present in horses, swine and ruminants. Then there is an endotheliocorial. So, when the parturition takes place in such animals, there is no bleeding that occurs, because it kinds of just comes out easily from the fetal tissue, comes out easily from the maternal tissue which is there. Then there is termed as endotheliocorial, where the digestion of the where the digestion of the epithelial cells have taken place, endometrial epithelium has taken place, and the connective tissue has also been degraded, but the uterine endothelium remains intact. So the three layers of fetal tissue is intact, but only one layer of the maternal tissue is there, and this is present in case of dogs and cats. Then there is a hemocorial placenta. In hemocorial placenta, the uterine endothelium also breaks down, and that is when I said that the fetal tissue is actually bathing in the maternal blood, and this is present in case of humans and rodents. So when the parturition takes place, when the fetal tissue comes out, then there is a bleeding that has occurred, or there is a shedding of the endometrial wall, and therefore the blood also comes out, because there are ruptured vessels which are there. So in this case, the uterine endothelium is there. So based upon which maternal layers are retained histologically, there can be epitheliocorial placenta, endotheliocorial placenta and hemocorial placenta. So these are the feto-maternal interdigitation that occurs, and different types of placentas that are formed. The human placenta is therefore an allantochorial placenta. The chorial placental circulation is connected with the fetal allantois. Hemocorial placenta, because when the parturition occurs, then there is a blood release, so hemorrhage occurs. It's discoidal because there is a discoid kind of placenta arrangement which is there, and pseudocotyledonary because the villi are grouped and incompletely separated by walls that are between them. So they do form groups, therefore it is discoidal because it is circular, still circular in structure, and it's in a disc form. But cotyledonary because on the surface these villi create the impression of cotyledons, although they are not completely cotyledonary. So we can say that it is a pseudo-cotyledonary this thing, and decidual placenta. So again there are two types of placenta on the basis of the material it takes out during the parturition, the decidual placenta and the non-decidual placenta. So decidual placenta is a placenta that takes part of the uterine mucosa with it when it is expelled. However, non-decidual placenta are placentas which do not take the maternal uterine mucosa along with it when the parturition takes place. So it happens in the case of cows, etc., where the two tissues are just lying in between each other, and when the parturition occurs, they separate without the loss of either of the tissues. So fetal tissue just separates from the maternal tissue without the loss of maternal tissue. But when the maternal tissue loss occurs, then it is termed as the decidual placenta. So human placenta is allantochorial, hemocorial, discoidal, pseudocotyledonary and decidual placenta. Coming to the functions or the physiology of placenta, the role of the placenta in the feto-maternal exchange processes. The placental exchange processes occur via classic membranous transport mechanism. So the passive transport can take place, which is without the energy consumption. So there is simple diffusion. The non-polar molecules and the fat dissolvable substances follow the concentration gradients. They diffuse from the side with the higher concentration to the side with the lower concentration until a balanced condition is achieved whereby no energy is used up in the process. For example, the diffusion of oxygen. So various gases. So the movement takes place by passive simple diffusion. It can take place by osmosis, the diffusion of the soluble substances through a membrane with selective permeability. Water, a strongly polarized molecule, cannot penetrate through the double lipid layer of the cellular membrane, but it crosses the placenta through specialized pores which are termed as the aquaporins or water channels, the proteins localized within the plasma membrane. Then there can be simplified transport, which is transition from the side with higher concentration to the one with lower concentration with the help of transport molecules. So the exchange of materials between the fetus and the maternal side can take place just like any membranous transport that can occur, any exchange across the plasma membrane that can take place. So by passive diffusion, the simple diffusion, osmosis and simple transport, as well as they can be transported by an active mechanism where the utilization of energy takes place, and there are which can take place against the concentration gradient, and there are pumps such as sodium potassium ATPase or calcium ions are required. Then there can be vesicular transport, the endocytosis or exocytosis across the placental membrane also. Macromolecules are captured by microvilli and absorbed in the cells or repelled, such as the immunoglobulins or the antibodies are transferred from the maternal tissue to the fetal side by the receptor mediated endocytosis, and this is how the transport can take place across the placental barriers. The various functions which the placenta takes up is the respiratory function, wherein the respiratory function makes the fetal oxygen supply and fetal carbon dioxide removal possible. So oxygen from the maternal blood is taken up and the carbon dioxide is removed. The placenta, which plays the role of fetal lungs, is 15 times less efficient with equivalent weight of tissue than the real lungs. The supply of the fetus with oxygen is facilitated by three factors: the difference of oxygen concentration and partial pressure within the feto-maternal circulatory system, higher affinity of the fetal hemoglobin for oxygen, and the Bohr effect. So basically, dependent upon the concentration and the partial pressure of the gases across the membranes, one gas can be loaded and the other gas can be downloaded at the junction or the placental barrier, and that's how the respiration takes place, or that's how the fetal tissue gets its oxygen supply from, or the removal of carbon dioxide also occurs by that mechanism. Then various nutritive and excretory functions are also taken up by the placenta. The water diffuses into the placenta along an osmolar gradient through the aquaporins. The water exchange increases during the pregnancy up to 35th week, which is like 3.5 liter per day is transported. The electrolytes follow the water, whereby iron and calcium only go from the mother to the child. So electrolytes, they go from the water, and the iron and calcium are also going from the mother to the child. The glucose is the fetus' main source of energy and passes the placenta via simplified transport. The fetal blood sugar concentration is 2/3 of the mother's and depends upon it. So glucose is also transported through the placenta. Proteins are too large to pass through the placental barrier. However, the peptides and amino acids can pass through via active transport and thus ensure the fetus' own protein synthesis. So the raw materials can pass on from the maternal side to the fetal side, where the protein synthesis can take place. So proteins are large enough to cross the placental barrier and therefore they are not transferred. The amino acid precursors of fetal protein synthesis stem from the metabolism of the maternal proteins. The placental transport is facilitated by the influence of hormones such as the growth hormone and the thyroid stimulating hormones against a concentration gradient, because the concentration of these material are higher in the fetus than in the mother. So 2 to 3 times higher in the fetus. Then across the concentration gradient, by the active transport, these materials are transported into the fetal system, where the synthesis of the proteins can take place. Then lipids and triglycerides are decomposed in the placenta, where new lipid molecules are synthesized. So decomposition as well as creation of the new lipid and triglyceride molecules can occur in the placenta. The cholesterol passes through the placental membrane easily, just like its derivatives, for example, steroid hormones. Water-soluble vitamins easily pass through the placental membrane. The amount of the fat-soluble vitamins, which is A, D, E and K in the fetal circulation, is on the other hand quite low. So vitamin K plays an important role in blood coagulation and is applied to the child immediately after birth in order to prevent hemorrhages. So basically, fat-soluble vitamins, they are low in concentration. However, water-soluble vitamins are higher in concentration in the fetal tissue, and they can easily pass through the placental barrier. The placental exchange processes are also involved in the removal of waste products from the fetal metabolism. They cross over into the maternal blood in order to be excreted by the mother in the form of urea, creatinine or uric acid. So, the excretory system of the fetal tissue, the waste that it creates, this waste also passes from the fetal side to the maternal side through this placental barrier and is finally getting thrown out of the body. So, the placenta also carries on various nutritive and excretory functions. The placenta also acts as an immunological barrier. During pregnancy, the mother developed a tolerance to her child. This phenomena is based on the specific antigen property of the embryo and the placenta, as well as on the transitory changes of the maternal immune system during pregnancy. A body does not accept any foreign substance to be there. When the baby is developing inside, then the HLA receptors that it has on its surface are different from that of the mother's, and therefore mother can reject it based upon this immunological impacts that can occur. So, this placenta also act as immunological barriers, preventing the maternal side immunoglobulins to cross through and pass on to the fetal tissue except for one immunoglobulin, and then therefore the tolerance for the baby is created and the baby is not considered as a foreign substance and therefore allowed to grow inside the mother's body. The placenta also gives rise to a protective function. Strictly speaking, the placenta forms a protective barrier against infectious agents. Nevertheless, however, there are some microbes that can cross this barrier occasionally following the placental lesions. So, certain sexually transmitted diseases, for example, if a mother has an HIV, then that HIV virus can pass through the placenta and the fetal can also develop the HIV infection, can also have HIV infection. Then other viruses such as rubella virus, these viruses, or the Mycobacterium tuberculosis virus, these viruses can also pass on from the mother and can cross the placental barrier and goes into the fetal tissues. Apart from that, the placenta also presents an incomplete barrier against certain injurious effects of drugs. Antibiotics and corticoids can pass through the placental barrier. Depending on their size, certain steroid hormones get through as well. So, certain drugs such as thalidomide and etc., or the effect of alcohol also can reach to the baby, the growing baby, and can pass through the placental barrier and can affect the baby. The endocrinal function of the placenta and especially the syncytial trophoblast can be seen as a large endocrine gland. At the beginning of the pregnancy, the synthesis of estrogen and progesterone is ensured by the corpus luteum gravidatus that is maintained by the human choriogonadotropin HCG, a product of the trophoblast. The activity of the corpus luteum decreases progressively with the beginning of the 8th week in order to be entirely replaced by the placenta at the end of the first trimester. So, initially the placenta at the beginning of the pregnancy, this trophoblastic layer releases this HCG, and therefore by the end of first trimester, this hormonal secretion is taken over by the placenta. During the pregnancy, the hormonal concentration in the maternal blood is regulated by the cooperation of the placental, hypophyseal and fetal suprarenal hormones as well as hormones from the gonads. So, endocrinal function is also performed by the placenta. So, we have seen that the growing embryo basically is implanted into the uterine epithelium. That is the conducive environment where the development can take place, and for the development to occur, this implantation, the proper implantation is very much necessary, and implantation occur into by different there are different phases of implantation such as the apposition, the attachment and the invasion, and finally this growing blastocyst actually invades the endometrial wall, comes to lie underneath it, and the development goes on. the formation of a structure, a transient structure through which the exchange of material between the fetal and the maternal tissues take place, which is called as the placenta, and through this placental barrier various respiratory exchanges or excretory exchanges as well as nutritive exchanges takes place, giving nutrition and helping in the different functions of the growing embryo or the growing adult, and then the development can take place. So, this is all about the implantation and placentation, and we will move on to the next topics in our subsequent sessions.
Dear friends, on that note, we would like to thank ma'am for this very wonderful lecture, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.